Thanks for the introduction, Dr. Rovia, and thanks everyone to coming this session. My name is Zhen, and I'm a fourth-year student at the University of Minnesota, and also second-year master student. So it's my honor to be here to share some our understanding of PCV3 from our previous one-year study. And first of all, we'll talk a little about the identification of this virus. This virus was first reported in 2016 from two research groups in the United States, and then also later reported in many countries in North America, Asia, and Europe. And we'll see the list. This list of countries found the positive cases in the red colors. It just continued to grow. So, but when we go back to histological samples of different countries, we found this virus has been found in earlier than 2016. For example, in the United States, when we go back to the sequence data, they found they have a partial sequence of PCV3 in the pork meat. And also in the UK, they found their archive samples have PCV3 positive cases back to 2002. In Spain and China, they found their case in 1996. And recent paper in Sweden, they found this virus in 1993. So the conclusion is this PCV3 may have been circulating in the swine population in different countries for many, many years. And we just found this virus recently. So when we studied the PCV3, we were always asked, what's the difference between the PCV2 and the PCV3? So genetically, they are all single-strand circular DNA, and the PCV2 has about 176 kilobase with two major open reading frames. And ORF1 encodes replicase, and ORF2 encodes capsid. And based on the ORF2 genes, which encodes capsid, the PCV2 can be classified in 5 different genotypes from PCV2A to PCV2E. And also, we have lots of commercial vaccines for the PCV2 right now. As for PCV3, the genome is a little bit bigger, about 2 kilobase. And then there was 3 open reading frames has been predicted. And we have no good criteria to classify those the PCV3 virus in different genotypes. And one reason is this virus genome is very similar. The others, we don't have a criteria. We cannot use the same criteria of PCV2 to apply to PCV3. And so far, there's no commercial available vaccine for PCV3, and we haven't heard any success in any laboratory reported they have a virus isolation. So that's also the challenge to prove Cox postulate if this virus cannot repeat the clinical sign and isolate the virus in the end. So we look at the genomes of PCV3s, comparing the genome of PCV3 with other circovirus in other species and the mammalians. We found that PCV2 and 1 are close together with the 20% identities in the whole genome. But the PCV3 virus was right here, which shares less than 50% nucleotide or amino acid identity with PCV1 and 2. So this information can suggest that the current PCV2 vaccine, in theory, can now cross-protect a PCV3 virus due to their high genetic variation. So we compare the clinical signs of PCV2 and PCV3. We probably know that PCV2 causing the post-weaning multisystemic wasting syndromes, which AKA rapid weight loss, and porcine dermatitis necropathy syndromes, reproductive disorders, and respiratory disease. There are a few literatures has reported PCV3-associated clinical signs, including cardiomyopathy systemic inflammation, PDS, reproductive failure, acute death, and respiratory disease, PRDC, and congenital tremors. I put those plus and minus. It's just my thought how confident I have those in literatures that reporting those clinical signs. Because now all the literatures have fully diagnostic workup, there's some pathogens haven't been ruled out in their case report. But if you attend the seminar yesterday from Dr. Bayer, they recently found congenital tremor cases, and they showed the clinical lesion in the brains as well. So histologically, PCV2 are associated with lymphoid depletion, macrophage infiltrations, and a few reports found PCV3 may causing myocarditis. And also these slides from Dr. Rovia, you can see the heart kidney, and spleen, they all have the vasculitis. And this picture on the left is the in situ hybridization to show the virus particle in the tissues. So now the big questions right now in the industry is the PCV3 a real threat to our pig industry? So they think about PCV2 about 40 or 50 years ago. It was found in very early time in 1970s, and then it spread out in Europe and causing problems in 1990s to 2000, and later on come to US in 2006 and 2007 and cause a huge economic impact in our swine industry. So will the same story happen in PCV3? I bring some opinions from the practitioner and diagnosticians and scientists. Dr. Bayer said, if the PCV3 is identified in swine with clinical PCVD and the histopathology consistent with circovirus infection exists in the absence of PCV2, then the PCV3 is an important disease-causing pathogen. But still, she still believes that in some situations, the PCV3 can be the primary pathogen. So the research papers from Spain, their data suggests their exploratory study is now in line with the putative role of PCV3 as a major threat to the pig health. And I sent emails to confirm to Dr. Zhen from Iowa State, and he said it remains to be determined whether PCV3 is a causative agent by itself. Experimental study are needed to confirm whether PCV3 alone can cause a disease or other factors such as co-infection and environmental factors are needed. And I've also bring the opinion from Dr. Rovia is PCV3 appears to be a pathogen, but most infections result are subclinical. So now to bring our study, so we want to understand the geographic distribution, prevalence, clinical relevance, and the genetic diversity in the PCV3 in our diagnostic lab. And specifically, we want to understand the frequency of PCV3 in their geographic distribution, age, and viral co-infection, particular PCV2 and PERS. And also, we want to dig more further to collect those clinical signs and lesions to understand is there any association between those clinical signs and lesions with exposure of PCV3 virus. And also, we sequence a few samples to get a full genome sequencing to understand the genetic variation of PCV3 genomes in the United States. So, our study population is 2,177 samples with a PCV test from Minnesota Diagnostic Laboratory from February 2016 to January 2018. And those samples, initially we have an in-house qPCR, and then later we use a QIAGEN viral type PCV2 and 3 duplex after November 14, 2017. And we have the descriptive analysis of the risk of PCV positive samples, cases and size, as well as the PCV3 geographic distribution and age distribution. We collect those clinical science reports to analyze those clinical signs described by veterinarian and diagnosticians with the PCV test. And also we extract those histological lesions in those pigs and do a statistical analysis to see what's the association between those each individual lesion to the PCV3 infections. And we try the next generation sequencing and the secondary sequencing to sequence a few samples in our study. So the first result I'm going to show you is the PCV3 geographic distribution, the PCV3 positive rate. In our samples, 27% of samples were positive, and in cases were 35%. In the sites, different farm sites was 38%, and in the States is 86%. When you look at this graph, the yellow ones are positive state with PCV3, and the green one was a negative, and the green one was a sample. We don't have a sample from those states. So when you look at those states in the green, they don't have many pigs, and also we don't have many samples from those states as well. So it could be a false negative in this map. When we look at the PCV3 co-infections, we look at two viruses, PCV2 and PERS. In our samples with PCV2 test, the positive rate of PCV2 is 14%, and the co-infection with PCV2 and 3 was 1.2%. But we'll make this 2x2 table. You can see the odds ratio of PCV2 exposure versus the PCV3 outcomes was 2.2, and it is statistically significant, which means that the PCV2 infections have association, is associated with the PCV3 in our study population. As for the PERS, We have 38% of positive rate in our samples with the PERS test. The co-infection rate was 7.6%, but the odds ratio is less than 1. So we didn't find an association between the PERS and PCV3. So in our specimens can be found in PCV3 as followed. We have a tissue homogenates, which is a combination of lung, heart, kidney, spleen, and lymph node. We found PCV3 in the serum, blood, oral fluid, and processing fluid. In other tissues, such as individual lung, heart, lymph node, sexual gland organs, bone marrow, spinal cord, intestine, and feces. So majority of samples are from tissue homogeneous serum, oral fluid, and processing fluid. The oral fluid and the processing fluid have a higher rate, as it can be because there's a pool samples, and this sample type is more sensitive than the other sample types, such as tissue homogeneous or serum. When looking at the age distributions, we found the PCV3 from the fetus to the finishing, and which is about 15 to 20%. We have a higher rate in the adults, which mostly sows and some boars. And we have a huge number of samples have unknown ages. So once I figure out the distribution age, this category can definitely change the distribution age of PCV3 infection. But so far, this work haven't been finished yet. So when we look at the clinical signs, there's 10 category clinical signs. I extrapolate those diagnosis reports, which from abortions to weight loss. And we have 6, so these are the case numbers, not sample number. So each case, the veterinarian may have a description of this case, for example, abortion. And then we calculate those information and classify with PCV3 positive cases and versus PCV3 negative cases. And we found a higher rate rate in the abortion, which was about 40%. We also do a statistical analysis to try to figure out any association between PCV3 infection or clinical signs. And here's our data. So we'll look at the odds ratio for the abortions. The odds ratio is 2.4 and also statistically significant in the abortions. We didn't find the odds ratio beyond 1 in other clinical signs, and also the p-values do not support any association with PCV3 exposure. So now we're very interested about abortion cases. We found a higher number of cases has abortion and is associated with PCV infections. So what's the other co-infection may contribute those abortion cases? We'll look at them deeper to figure out their co-infections with other pathogens that can be contributed to abortion. For example, PERS, parvo, PCV2, etc. And then we categorize those individual infections and co-infection. So PERS have 7% of positive in those abortion cases. PCV2 is 6% and parvo is 13%. But once you eliminate the co-infections, we still have 11 cases that have PCV infections only, which may give some clue that PCV3 may have a role in abortion. So we have one case that have full diagnosis workup, and we have more information in this case. His whole mummies have PCV3 positive CT value 17. And we have a histological diagnosis of necrotic hepatitis, multifocal lymphocytic epicarditis, nephritis, and pneumonia. And interestingly, no other pathogens unknown causing abortion has been found in these cases. We go to deeper to the lesions. Here, there's the lung lesions in the aborted fetus. And there's the PCV3 viruses from the institute hybridization. When we go to the deeper, the red spots are PCV3 virus sequence. So this case gave us more ideas that we found a lesion in the aborted fetus that's probably associated with PCV3. And we didn't find other pathogen associated abortion in this case. So we want to figure out the clinical signs, but also want to understand the clinical lesions associated with PCV3. And then I went back to the same study population and read through those diagnosis report and try to categorize those lesion types by different organs and lesions, and try to understand any association with PCV3 infections, and also try to include those virus, bacteria, parasites, and age information for the future to do a more complicated model to try to figure out the association. But so far, we have a 2x2 table and also we shown the chi-score and Fisher exact test. So here's our data. So for the 16 organs and 38 types of lesions, we found the lesion in the heart and spleen and lung have a higher odds ratio over 1, and also they are all significant have statistically significant difference in the p-values. And the lesion types are myocarditis, heart vasculitis, and spleen vasculitis and interstitial pneumonia. So basically, multisystemic vasculitis plus interstitial pneumonia are the lesions we found are associated with PCV3 infection. Lastly, I want to touch a little bit more the genetic sequencing data we have. In this study, we got 1 complete genome from next-generation sequencing and 6 complete genomes from Sanger sequencing. So the sequence we got in this study is in the red, and the sequencing genomes from other studies from the US are green. So you look at these phylogenetic trees, the US genomes are now clustered together, and they are spread out in the phylogenetic trees. Some are more close to the strains found in Asia, and some more close to the strains found in Europe and Brazil. So, which suggests that this virus has been circulated for many years, which has been proved by many literatures, but also can be that this virus is not a single source introduction to the United States. And also the whole genome survival, the PCV3 genes in the US and other countries share over 97.6% of nucleotide identity. And in summary, we found PCV3 is widespread in the world, but also is widespread in the United States. And the co-infection of PCV2 is commonly found, and we think that is associated with PCV3 infections. There's also a higher rate in the PERS, but we didn't find an association between the PERS and the PCV2. So PCV3 can be found in different peg age from fetus to adults and in multiple specimen types. And abortion have a higher rate in our clinical science, and our abortion rate is higher, about 40% positive. And we have 1 case report of PCV-associated histological lesions or border fetus in the lung. So the lesions like myocarditis, vasculitis, and interstitial pneumonia were associated with PCV3 infections. And genetically, current PCV genomes in the US share over 98% identity. Well, at one more type of genome analysis, there's a lot of research group from other countries try to classify those PCV3 genomes in different genotypes. But one research study from Germany, when you do the genome comparisons, they use the wrong sequence data. So give you an idea, oh, there's a separate nucleate or genotype of PCV3. But when you're going back those NCBI, and then the original author has changed the sequence data, and we use a new sequence data analysis, that pattern just goes away. So that is my reference. And I will thank my advisor, Dr. Rovia, and my committee members, Dr. Matler and Monsignor Toromoro. And I will thank Dr. Fabian Vanucci, and also faculty and staff from Veterinary Diagnostic Laboratory. And I will thank the funding support from Veterinary and Population Medicine Department, Summer Scholar Program, Chinese Scholar Council, and the Swine Health Information Center.